You should go, perhaps, just in case, sharing.
Yes. So I'm really happy today. Our talk will be about morphogenesis. We have both Dagmar, Iber, and Sid Goyal. And just really quick, morphogenesis is not an easy word. It comes really loaded. Just trying to understand how shape grows across development and time, and just morphology in itself is already so complicated, has taken a lot of developmental biologists a lot of work to figure out how these things are timed in the right way. And as far as I'm concerned, Dagmar is one of the leaders in development and morphology and shape and form. And so I'm really happy to have her here. Oh, we can't hear you.
Right. Thanks for organizing this. I mean, I think it's a really fantastic initiative. I was saying this before. So very happy to do this and have the opportunity to talk about our work or introduce it briefly. From what I understand, it's like 25 minutes overall, but like it's meant to be interactive. So you're welcome to interrupt at any moment. I only have one screen, so I might not see it, but just stop me and talk over me. So what my group is interested in is really the principles of development. So the basic question is like, how can a complex organism with complex functionalities develop from a single cell? And just see, it really starts here. So when I came out of my master studies, this paper just got published, and the newspapers, and anyone who has lived in those days will know what I mean. The newspapers were full with statements that the book of life had just been deciphered. And as we know by now, as we don't understand this book very much, we have identified all the protein coding genes, but how they're translated into a 3D organism is still largely elusive. And so that's basically the question that motivates me and my group to understand how to make sense out of this piece of information, and in particular, how to translate this linear string of information into 3D shapes and functionalities. And that means we're interested in the fundamental questions in developmental biology. One very important one is pattern formation. How are patterns encoded? How are they controlled? How do you get precise patterning? And also very important, related to that, how do you break symmetries as you develop life? Then how do you coordinate processes during development? How do you control growth? How do you terminate growth? And then finally, how do you evolve mechanisms? And most people, when they think of evolution, they think about generating new functionalities. But much as you have to develop new stuff, you also have to preserve the old stuff. And you have to preserve it even though you're changing the size of your embryos and the developmental pace. And that is just very difficult. And so my group, as I said, are interested in fundamental mechanisms. One is patterning. And so we have worked on morphogen gradients for a long time. We have asked questions about how they encode position on a growing domain. So you all know the French flag model, I suppose. You have a concentration gradient. Then you have concentration thresholds, and they encode boundaries within a tissue. And you get different cell fates in these different sub-regions. And we have worked also on scaling, means like when you have embryos of different size, how would you still maintain the same proportions? And another aspect that has come in the last couple of years for us is like the 3D organization of epithelia. So like most of the patterning is happening in epithelia, and here you're just seeing a cartoon of these cells, but in reality they're really they're having a really complex shape. And if we want to simulate tissues in a good way, we need to understand the biophysics of that organization and also what it implies or means for patterning. And so in this case here, we realize there's like 14 neighbor changes from top to bottom, from apical to basal, and we could explain those neighbor changes with energy minimization, but also means these cells have enormous number of contacts, many more than one would expect based on the surfaces. And then we have worked on growth control for a long time. And here's just an example of the Drosophila eye disc, where we found that dilution might explain the decline in growth rate and final growth termination, and dilution as the disc is growing. And beyond these fundamental mechanisms, we are interested in what we call in silico organogenesis. So what we're doing here is that we're obtaining 3D shapes of organs, in particular here's lung and kidney, and then we're trying to explain morphogenesis. And one process we are particularly interested in is branching morphogenesis, and here we have been able to explain branch point formation with the Turing mechanism based on established growth factors. Then we're interested in the coordination of processes, as I said. Here you're seeing an example of a kidney. In green, you're seeing the forming ureter, and in yellow, you're seeing the forming nephrons, which are giving rise to the filter unit of the kidney. And these two processes really need to be tightly coordinated. So each branch point needs to have one nephron forming, and it's been an open question how this is coordinated. And people had ideas about pre-patterns, and what we now show is that people had rejected geometric effects, but what we are now showing is that geometric effects really explain this coordination. Means like you have one protein that is expressed in the entire ureter in the green part, and then in those armpits, in those corners, you get an up concentration of that protein, and that defines the positions of the nephrons forming. And then we are also interested in the development of the central nervous system. Here you're seeing the formation of that neural tube, which gives rise to the central nervous system. And this is this structure here. So you see how it's folding. And here for a long time, people believed this folding would be controlled by localized apical constriction, and this localization would be controlled by morphogen gradients. And what we showed is that you can explain it based on differential growth. So the area down here, which is the mesoderm, is expanding, and that expansion is pushing the neural plate up and allows it to close. And you also have to pin it down here by binding it to the notochord. And so that was a new mechanism that explained a lot of different aspects, but it also illustrates the difficulties that we have these days. And that is, we have a lot of candidate mechanisms that people have figured out over the decades. And now it's our job to not only recapitulate processes that we are seeing in vivo, but also distinguish which of these mechanisms is actually plausible. And this is only possible if you have a sufficient resolution. If you're just making conceptual models, you won't be able to do this test in a good way or in a reliable way. And that's why we're doing this image-based modeling that we have spent a lot of time and effort on. Now, today I want to focus on one aspect in particular because it's easier to convey via the screen, and that is about the precision of these gradient-based patterning mechanisms, which is one of the aspects that we're working on. So as I introduced before, we have this French flag model, according to which you have an exponential gradient, and then these concentration thresholds define boundaries in a tissue. And so that now we have to ask, what do we mean with precision? There's two different types of precisions. One is the precision of the transition zone, the sharpness here, which I'm not going to focus on today. And the second form of precision, which we also refer to as positional error, is the differences between individual embryos. And so that means if you have small differences here in those gradients, then these concentration thresholds will occur at slightly different positions within the domain. And that's gonna lead to shifts between embryos, and that's the positional error that we're interested in. And now the question is whether these chemical gradients can be sufficiently precise to provide reliable information during development. And we have looked at that, motivated by work that has previously done by the Briscoe group in the neural tube. And what the Briscoe group had published in 2017 in this paper by Zagorski et al. Is that they looked at the neural tube and they looked at the position of two boundaries here in the center of the neural tube. And when they looked at the precision of that readout, they found that the standard deviation of that position is only 2.5 up to 2.5, 3 cells, so quite precise. But when they quantified the morphogen gradients that should encode that position, then they found that in that part of the domain where you have this high readout precision, you have a very low gradient precision. And you can see the error here, up here, it exceeds 30 cell diameters. And so then the question was like, how could you get precise readouts nonetheless? And they at that time proposed that it would be 2 opposing gradients, and that the combined information in these 2 gradients would allow you to read out those gradients reliably. And when we analyzed the data, we realized there was a mathematical problem with the analysis, and that is the readout precision they had quantified by really looking at those different readout points and calculating the standard deviation. And that's the positional error. But for the gradients, even though they could have done the same thing, just define a readout threshold and then get the readout positions, they had used a different methodology. That is, they had approximated that by calculating, by using the slope of the average gradient. I don't want to go into the subtlety here now. You can find in our paper, or I can comment on it later. But just mathematically, if you assume that you have perfectly exponential gradients, then even then you're gonna get a huge overestimate of the positional error, exactly in the part of the domain where they find this huge error. So like we can explain that huge deviation already simply because of this mathematical inaccuracy of the approximation. And a second thing is that when they imaged the gradients, and it took a long time, but eventually they confirmed, they used 8-bit imaging. Now 8-bit, 2 to the 8 is 256, means they can only capture a 250-fold decline in signal. And so that means you can only get the gradient up to this 8-bit limit. So from the source, once you have an exponential decline in signal, and once the signal has declined by 250-fold, you just have white noise because your detector can't detect it anymore. And that's what you're seeing here in action. So they can only follow the exponential gradient close to the source, and then you're getting really white noise. And the positions that they had tried to analyze here are lying in this white noise area. And so that means, sure, you could use 16-bit imaging, but it meant the data just has technical problems. And so we, as computation people, we decided to take a different route and use a simulation framework to estimate what level of precision we would be able to expect from a morphogen gradient given molecular noise. So what are now the noise parts that we have to consider? So if you have a morphogen gradient, the morphogen gradient comes about because you have transport by diffusion, you have production in the source, so you're going to have a couple of source cells, or here in the domain you have source where the morphogen is produced, and then you have a patterning domain. In this patterning domain you only have diffusion and you have degradation. And now each cell is going to have a slightly different production rate or different degradation rate or different diffusion coefficient. And now the variability has been, so the mean and standard deviations, they have been determined at least in cell culture. So we know the CV, the coefficient of variation. And so we used log normal distributions with that coefficient of variation. And then we drew those parameters for each cell from that distribution and ran simulations. And as a consequence of the molecular noise levels, you're going to get variable gradients that differ from embryo to embryo. And based on that, we could then ask how variable, how different are they for molecular noise levels. So we're really using the measured noise levels. And when we do that, we're getting this here. We're getting, so you see the CV value is about 0.3 here. And so as we move further from the source, the positional error increases. That's to be expected. But you can see the positional error is less than about 3-4 cell diameters, even at a huge distance from the source. Now the PAC3 and NKX61 domains, they may be around here. And so that matches quite well what has been reported. And so that means that these readout precisions are absolutely consistent with what you would expect for a normal noisy morphogen gradient. And what comes on top is that the size of the progenitor domain, so what you're trying to do with a neural tube is like define progenitor domains, and these progenitor domains give rise to different neuron types. So for instance, motor neurons and so on. So if you want to have the right number of motor neurons, you don't care about the position of that motor neuron domain or the exact position, but only on its size. Now, if you're changing the source, if you're altering the source, you will shift both the start and the end of that domain in the same way. And so that means that we preserve the overall size of the domain. Then Nama Barkay, like 20 years ago, suggested that you could get even further increase in precision if you had nonlinear decay. So that means self-enhanced decay. When we test this one in our model, we see an effect, but it's really academic, as one could say. It's really tiny. So the effect exists close to the source, but further out, it also doesn't exist anymore. But the effect, as you can see, n equals 1, n equals 2, the effect is really tiny between linear and nonlinear decay. But what does help a lot is if you have a 2D domain, means like you don't just have one line of cells, but rather as here in the wing pouch, the Drosophila wing pouch, you have a certain width of the domain. And so if we have a single cell, then the positional error, as you can see for physiological parameters taken from the Drosophila wing disc, is above 1 cell diameter at later stages. But if we have 10 cells in a row, then it is maintained at less than 10 cell diameter, then less than 1 cell diameter. You can see here, if you make it broader than 10 cells, it doesn't really improve much anymore. So 10 cells is a perfect patterning width. Another thing that Jan realized is like if you have really small cells, then you have a higher patterning precision. And we notice that all the mean apical cell areas are small in tissues that are patterned by gradients and not in those that are not patterned by gradients. And so we concluded from here that it's beneficial to have slim cells. And indeed, what you're finding, and so far no one had an explanation why, in all those gradient, all tissues that are patterned by gradients, you have pseudostratification, means the cells are so thin that the nuclei are not all fitting in one row, and so they're dispersed along the apical-basal axis. And so far, no one had an explanation for why you would have this architecture, and people believe that precision might be the driving force, the evolutionary driving force. And so that means, as a consequence, as I said before, because you have pseudostratification, you have these neighbor changes, and so you have this red cells also has the blue cells as neighbor, which is meaning it has more interactions, but also you overestimate the positional error because the cells are shaped in this way that this nucleus here, you know, when you analyze this as Briscoe group did, you would think there's a positional error when the apical site is actually in the right position. And so that means that the positional error of 2.53 cells is probably overestimated. So we think patterning is probably more precise. And as I showed, it could be subcellular. So it probably is, it's really like ultimately precise, means like at a single cell position. So to sum up, we believe that gradients are a lot more precise than people previously thought. The molecular noise levels are clearly low enough for precise patterning, means like with a single cell resolution. And slim cells are important for that precision. And so the prediction we have, one would need to test this experimentally, is that if you make the cells wider, if you break the skew stratification, you wouldn't have as precise patterning. So just my question here, I think I'm already out of time. Is that correct? Maria?
You're okay. If you have just a few more minutes, I think we can.
Okay, okay. So then I would just
Okay
like, very good. So because of that, and also for other reasons, like we have an interest in cellular resolution. So like to really simulate tissues at cellular resolution. I showed before that we had this 1D model.
Can I ask a question? Sorry, can I ask a question, the previous part?
Sure.
So just to understand, so I guess the earlier work in Drosophila, where one was also talking about these precision things in embryos, we're looking at, beak wide profile and things like that. Again, similar sort of answer. Am I correct to say that the similar answer was there, that the gradients were actually kind of precise enough to do?
Yes, because it's a slightly different situation because you don't have cells. So like what we're working on.
Yes, you have nuclei though, right?
Exactly. They only have the nuclei, but like you also have diffusion between the nuclei because you have the syncytium, while we don't have that. So they had Thomas Krieger has this argument, people haven't yet confirmed it, that you would have increased precision because of this diffusion, because of the diffusion of transcription factors between nuclei, which leads to some averaging. So that's an additional effect, but what we would be saying, you don't need that in order to get the precision that is observed, especially because the nuclei are even smaller than the cells that we're looking at least as small.
I see. So the statement here is really that the gradient is not noisy. That's the statement you're making, that the gradient is just not noisy.
It's not as noisy as people thought it is. I mean, there's obviously noise in it, but it's a lot less than people always anticipated. And they thought that because they basically changed this gradient length lambda and C0 in a deterministic way. But if you vary it from cells to cell, then you have the law of large numbers. So it basically equilibrates or equalizes, averages out. And that's why the final variability of the gradients is a lot less than what people thought it would be.
I see. Okay, so basically you're saying it's the averaging. Okay, I get it. And experimentally you see that too, that the gradient doesn't have that much noise.
Experimentally they have a mess. That's what I showed, right? It's
right.
Very,
the readout was kind of a mess.
Any of these protein things. I mean, up to Thomas Krieger has done the best work out there in quantifying the Bicoid gradient, which is the easiest system to study. Any other system like the neural tube or like also Drosophila wingdis is much harder to study.
So have people measured the gradients here, or
yes, people have measured it, but it's very difficult because you have so many errors, technical errors when you do that, because any kind of system or any kind of fluorophore introduce a lot of errors. Yeah.
Got it.
So it needs to be remeasured. Yeah.
Thank you.
Well, just very briefly, so we are developing the cell-based tissue simulation frameworks. We initially had LBIB cell, which is has high cellular resolution.
Dagmar, I had a question, sorry.
Okay.
On the equation that you wrote down for the concentration profile as a function of time, do you assume that cells are fixed in position and also that there is no division happening?
Yes, at the moment for that analysis, we did do that. You could, of course, change that. You could have it on a growing domain. My PhD student is thinking about it. And we could integrate it here, right, into these kind of frameworks. So but this was a very simple 1D, and then also the 2D framework was really just a checkerboard. While here we have all the geometric resolution that one might want to consider, including cell division and growth and everything, right? So we could now reanalyze this in this setting. You're absolutely right.
And so for the Zagorski paper that you're showing for the precision and the error, is there a lot of division and cell rearrangements that are happening in that work?
That rearrangements not. I mean, that has been analyzed by the Briscoe group. There is some growth, but the gradients, I mean, so the timescale, the timescale of the growth process is so slow, you have a steady state gradient. I should say this, like all the analysis we have done is with steady state gradients, because they're clearly in the neural tubes, they're clearly in steady state. So the growth process is not so important in that scenario. Yeah.
Okay, thank you.
Right, and so we now have a really efficient platform where we can grow tissue in 2D to 100,000 cells within like, a day or so or less. I mean, very, very quickly. We have the same now in 3D. Here it takes about a week to have 100,000 cells, a day to get 30,000 cells, so from a single cell to 30,000. We have a huge geometric resolution. So you see this here, we can implement all the forces. So each cell is really meshed at high resolution. We can have vesicles, spheroids, sheets with the nuclei inside. We can grow the tissue. Here we can get multi-layered epithelia, single-layered epithelia, study all those impacts and conditions. We can, because of the high geometric resolution, we can segment microscopy images, pull that in, and use that to simulate, for instance, organoids or whatever tissue you want to simulate. So here, for instance, the 3D epithelia, we could extract the 3D geometries, very complex cells. In the blue cells, we fix, the red cells, we did the parameter screen. And this way, we could infer the biophysical parameters that lead to these really complex cell shapes. And what was really absolutely surprising to me is that we can use the same parameter set for all the red cells to reproduce their complex shapes. And that tells us it's very simple physics that defines their shapes. And we had said this all along, because when we did the analysis, when we first looked at it, we realized that in each plane, from apical to basal, even though the polygonal surfaces here look different, or slices look different, as they all follow the same fundamental laws, means like Above-Weier's law, Lewis' law, Euler's theorem. Also, we had a new relationship about the cellular dispersity, and we can explain these laws with energy minimization. It's basically the cells always try to assume the most regular polygonal shape, but given that those areas, cross-sectional areas, vary from cell to cell, this is achieved if the largest cells has the most neighbors. We can derive that, and also because you have to always have 360 degrees in at each vertex, while each polygonal type has a different preferred angle for regular polygons. You're getting a trade-off, and that leads to Above-Weier's law. So it's basically very simple energy considerations that lead to these fundamental phenomenological relationships. And so to sum up on this part, now with light sheet microscopy, we can get really high resolution images of these epithelia. With that, we could explain these really old, long-standing open questions, and explain with that the 3D epithelial organization. That means we now have a very faithful and high resolution 3D software that allows us to really recapitulate the biophysics of these tissues. And now I think I'm really coming to the, I have really gone over time, I fear. So I want to thank my group, of course, who have done all the work, our collaborators, and then, of course, you for engaging and listening today. And while I'm taking questions, I still want to show that opening, because I really struggle to find a postdoc for that one. That is really because I need someone with particular skill set, and maybe you know someone, or maybe the person is even here. I need a mathematical modeler, not a mathematician, not a biologist, but someone who's bridging the two, means who's understanding biology and can develop mathematical models. And we have clinical data sets from in vitro fertilization patients, and we want to understand the formation of the oocyte to help in the clinic. So it's a really nice personalized medicine, but really fundamental biology grant, human biology. So it fits perfectly with this series here. So thank you. And I should say, unlike in many other countries, postdocs in Switzerland are well paid, so you're getting like 90K, so $90,000 as a salary. Okay, good.
Thank you. Thank you, Dagmar. This was great. If it's okay, we can start with Sid just to get the presentations, and then if questions arise, we're going to have a discussion. So the plan is to have a longer discussion.
Okay, very good. So I'm just going to stop
Yeah, so as Sid is preparing, if you have questions, please ask Dagmar, but I just thought we could just continue with the presentations because
Can you see it? Can you guys see it?
Yes.
Okay, good. Sorry, I'll stop until.
No, it's great. Sid, this is great. And I'm really happy that also Sid agreed to give a talk here. For me, Sid is the go-to guy for any model. If you want to try to explain any of your uncertainties or errors or your data, he's really the person to talk to. And I enjoyed meeting him early on, especially as a theoretician. And I didn't have other modeling friends, so he was really my favorite person to go and talk to and just chat about ideas. So it's really great to have you here, Sid.
Thanks. Yeah, no, I think Toronto has slowly sort of have more and more people doing theory things, and it's been fun to sort of talk to. And this community is sort of definitely a lot of fun. I guess I should remove the postdoc advertisement I had in the end because I don't think we can match the salary. But anyways, I should say Morphogenesis, and I'm glad that I'm talking after Dagmar because she's introduced almost everything that I needed. So I'll sort of dive into a very particular problem I had thought, and this is something new for us. It's in collaboration with a local group here in Toronto, Sevan Hupian. I'll say more names later, people who worked. And the question this is a broad thing we're interested in. And this is, I think, as Dagmar said towards the end of her talk as well, that there are basic physical principles behind these things. And that's sort of something we are trying to develop, defining these biophysical mechanisms for organogenesis in general. And, lots of people are working on these. And the idea was to integrate the molecules we know into sort of mechanisms which might be used again and again in different parts of embryogenesis and organogenesis. And, maybe we can do them in vitro if we understand sort of a little bit more biophysically what is happening, and we can swap away the molecules we need. So the problem I'm going to talk about today is something we found surprising, where just on this image you can see sort of this is the neural tube formation as was mentioned, and at the end of it basically, or somewhere towards where this thing is folding, it looks a very ordered structure, right? You're sort of creating order, and that's what development is about. In some ways, you create order, and we don't have to worry about thermodynamics because we are constantly injecting energy and lots of mechanisms to do those things. So we found an example where it looked like sort of fluctuation and disorder was actually enabling the morphogenetic event, and that's what I wanted to talk about. So the idea was, we have these various morphogenetic themes which seem to be happening again and again across embryogenesis. Cell aggregation is a classic one. Streaming and migration is a big one. Cell ingression is what I'm going to talk a little bit more, is another mechanism where reused many times. And of course, branching morphogenesis, as was also mentioned, is an important one where clearly, lungs and kidneys both do similar things. And it's happening even earlier in embryogenesis. And the idea was, can we think of them in some sort of physical language, right? And the motivation is twofold, not only to find what mechanisms might be going on, but also it might give us a way of, in the virtual human context, create things in artificial context a little bit better, right? One way to do it is replicate all the complexity of biology, get all the right molecules to interact with the right things. The other is maybe you don't need everything. You just need to figure out how cells are using the molecules to do run these mechanisms, and then we can sort of maybe get to in vitro problems a little bit in a different way rather than sort of a very molecular way. So let me go to the neural crest problem. So cell ingression is again not only happening in neural crest context, but there are sort of other places where cell ingression happens. Cell ingression I'm going to say more about, but roughly what is the idea is that there are two things happen at the same time. Of course, there's a physical movement of cells as they go from epithelia to mesoderm. And of course, in doing that, they have to do a epithelia to mesodermal, so EMT transition, mesenchymal transition. So this transition happens, and at the same time, cells are moving around, right? So this, in context of neural crest, this happens at, of course, it happens in, it's an important cell type, which is related apparently to the evolution of vertebrates. So you don't see these cell types before even sort of invertebrates, which are very vertebrate-like. But once you have vertebrates, you have these neural crest cells. And for example, they're extremely important in the context of neural crest to make all the craniofacial bones and lots of other things which are formed really from neural crest derived cells. So they really stream from epithelia. So here, just to point out things, so here, these green edges are basically where neural crest cells emerge, and they are between the neuroepithelial, so they're always at the boundaries, right? Interesting things happen at the boundary of sciences as well, and I think in biology also somehow this evolution happened at the boundary of cell types. So these neural crest cells that this cartoon shows in context of mouse is sort of, you know, these green parts is where the neural crest cells are defined. There are lots of genetic markers and things like that which define them. They have particular gene expression profiles, and then these cells sort of stream inside. They go into the mesoderm and form structures. So this is an image from Sivan's group. Just orient people a little bit. So here, this is sort of what, this is 4 or 5 somite state of mouse embryo. This is where the neural crest cells are on the edges. There are 2 different views. And here in the bigger image, these cells at the bottom are the cells which are sort of ingressing. So I'll try to maybe show it in a sort of a 3D view. And this is all thanks to Sivan. So these are the cells which are sort of ingressing now in. And the interesting thing is these cells start ingressing, and then they actually start coming together. So the cells delaminate, they come in, a few of the cells around them start delaminating, then they all come together and make a stream that then flows downwards, right? What I'm going to talk mostly about is this early part of delamination and the EMT transition. So what was surprising, at least the first surprise we had was, and this, if I understand correctly, and you know, people who know more should correct me, is that in context of things like xenopus, it looks like the migration is really driven by some gradients of stiffness and chemotaxis. Here it looked like the cortical fluctuations seem to be really central, and I'll just give you an experimental observation which sort of motivated this. Is so these are cortical fluctuations in neuroepithelial cells, which are the neighboring cells and next to the neural crest cells. And here the red lines are typically showing the fluctuations in the neural crest pre-ingress cells. So some of these cells start actually fluctuating a lot. The blue lines are going to be mutants, and I'm going to talk about it in a minute. But these fluctuations seems to be central. And if you actually do mutants of, you know, myosin motors, for example, this particular one, you can suppress these fluctuations. For example, the blue lines are corresponding to these mutants. And the 2 embryos here are showing you sort of that the percent of ingress cells is hugely reduced once the fluctuations are taken out, right? And of course, these are all pleiotropic effects. There could be more things going on. But we think fluctuations are playing one of the central roles in this process of allowing ingression to happen. And again, there's a catch-22 in terms of whether it's the EMT transition. Is it stopping the cell type transition, or is it stopping the physics of it in some ways? So that is something which we still don't have an answer to, but the 2 processes happen hand in glove. But clearly, if the fluctuations are not there, you don't see as much ingression. So this was sort of an interesting observation. We thought, can we see that in very simple models, right? And the idea was, you know, does fluctuation allow something to happen? And so here we were thinking of, you know, some axes like active fluctuations. And on the other hand, you have sort of this cell shape heterogeneity, which is important for cell fate transitions and sort of ingression and migration. So we had a bit of a cartoon of something like this, where, you know, you can generate ingression. And we sort of coded it into, this is a 2D vertex model. There was some thoughts of doing it 3D, and, you know, maybe we'll borrow frameworks which were presented to try on a 3D, but this is a 2D vertex model. Very simple. Again, the idea was to see conceptually if we can see something. And this is what we sort of cooked up in this particular context. So the idea is that if you take a green cell here, for example, if you can see, sorry, it's a little bit small, and you bring it closer to, so the blue and the green represent the epithelia and the red represent the mesoderm, right? So there's some property differences, they don't mix because of the addition between each other is different. But if you take a green cell, which is almost epithelia-like, and drag it down into the mesoderm, you see that deterministically it has to cross a barrier, right? So basically, as the cells get closer to the boundary, they actually don't like to go deterministically. They would actually come back. Then what we did in this very simple model was basically enhance fluctuations. And the fluctuations here are basically that the properties of the cells are different now, and the properties are very coarse. This is the surface to volume type ratio, which tells you whether they like to round up or they want to sort of flatten out, right? And as you enhance fluctuation, you basically see that this barrier crossing happens quite naturally, right? So what is happening is as the fluctuation in the epithelia is rising, they can actually ingress into the mesoderm, right? Of course, in this case, there is energy difference as well. So once they reach the other side, they can be stable, right? But this is not something we knew could happen in the real embryo, right? So we were curious what really stabilizes cells once they go into mesoderm. And again, there are many things that could happen. And this is just some of the images to highlight what is going on. This is the apical view. This is that cell, which is going to now sort of vanish, if you will, and just ingresses and drops down from epithelia to mesoderm. And you can see all the interesting things which Dagmar has already mentioned, the shapes of the cells. They are sort of, look at these 2D images. It doesn't quite capture the not only complexity, but also kind of interestingness of, how these cells are shaped and things like that. But you can see this cell sort of ingress. So we were curious, so one thing looks like the fluctuations allow these cells to move and cross barriers, which is again something we can appreciate from some physics language. But the idea was, what traps them? So once they go down, why don't they come back up? They're still fluctuating a lot, so they should be able to go in and out rather than being stuck one place or the other. So something is trapping them once they reach the mesoderm. And again, there could be many things, but one thing which sort of became very clear is that, for example, as Dagmar mentioned, the number of neighbors changes drastically in the mesoderm compared to when they're in epithelia. So on the left is basically, for example, neuroepithelia or the neural crest cell, which are just starting to ingress. They sort of have on average 8 to 10 neighbors. But by the time they reach in mesoderm, they basically have, 12 to 14 neighbors, basically. So many more neighbors. So our simple thought right now is that perhaps this sort of increased entropy, if you will, allows cells to get trapped in this new state, right? So the very simple model is that fluctuations allow things to move around, but moving around doesn't tell you which direction to go. They just basically everything moves around more. But then once they reach the state, which is in the mesoderm, something has to trap them. And we think that sort of, there's an entropic-like trap. Sorry, there should be an R in that. There's an entropic trap which captures the cells, which sort of enables cells to not go back. So in some ways, this is sort of a very, to me, I think it's an interesting picture where, it's sort of a mixture of randomness, which is really allowing this particular morphogenetic event to happen, which then leads to creating these streams, as I said. And the notion is that the streams then move because they have a gradient. Stiffness gradients are something which people have talked about, but there could be chemotactic gradient as well, which allows these streams of cells to move further down. But we don't think there are many, we have seen many examples where sort of fluctuation and entropic traps. It's a very non-equilibrium physics sort of idea where, you create trajectories to move around a lot, then you just trap them somewhere. And this is something which people have actually been thinking a lot about in active matter field more recently as well, but that's a whole different discussion. So we were quite excited about it. So this was sort of, as I said, work done in Savan's group, and, and I was helping out with the theory part, but it was a lot of fun to develop it. And this is sort of, it's on bioRxiv right now, and again, sitting at some editor's desk. So if you have any thoughts, feedback, we would be very excited. We think it's a little bit provocative the way we have said things in the paper, but I think there's something interesting, and maybe a general theme like that might we be able to see in other examples. So maybe I have a few more minutes. I'll just say a few more things that we do as a group, and I'll give a shout out to Spencer, who's in the audience. So she has been, so this is something else we have been doing in parallel, quite related to, I would say, self-fate dynamics more than morphogenesis, which at least in the previous example, this EMT transition is at the base of it. But the idea here was, can we learn the dynamical system which is central when self-fate transitions happen? And again, lots of people are working on it. And Spencer has done some really creative work in trying to infer these dynamical systems. This is just an example at the bottom of a simulated system where you can find these trajectories, how cells go from one type to the other. She has attempted this on various data sets. This is one of the examples of, endocrine progenitors going into these 4 terminal states, alpha, beta, delta, and epsilon. And here again, in a low dimensional sort of representation, she can now actually create these trajectories of going from, these stem-like cells to either beta cells or, for example, epsilon cells. And these trajectories, how they diverge, can provide sort of interesting places to see things around. She's extending this work to make it more predictive, in the sense, if you really started with positions which were a little bit further away because of genetic mutations, environmental perturbations, how could these trajectories then be predicted going forward in time? And this is again on bioRxiv, if people are interested in reading about it. I guess I should stop here and maybe give a chance for people to ask questions. I am not going to again talk about salaries, but we do interesting stuff. And some of this self-fate and morphogenesis is something we want to sort of put together in some way and think about those problems together, where feedback between the mechanics and the gene expression changes, again, something which lots of people are working on, seems to be central in many of these sort of morphogenetic events. So we want to think, I think what I want to highlight is we really want to think of events and think about, so important dynamical events which happen during morphogenesis, and how can we capture them in a language which might allow us to translate them into in vitro without really taking the whole molecular picture with us, right? So that's sort of our driving force in this case. And also, I think conceptually, I would put my money on having some sort of mechanisms which are translatable rather than molecules at times. Okay, I think I'll stop here and be open for questions.
Thank you, Sid. I'm actually going to try to open the floor so I can have a discussion with the two. Everybody can. I mean, I hope everybody appreciates. We just saw 2 different perspectives, both trying to answer shape and pattern formation. And I guess my question to both of you, because I think from Dagmar's talk, we kind of get the idea that some gradients is sort of directing or trying to help make development more precise, because that's kind of the idea of development. We're trying to control and get cells to show up at the right place at the right time. And then I also get the idea that you need sort of these noisy fluctuations in order to push yourself in and out of certain cell states. So I'm wondering if one way to just sort of join the 2 forms, let's assume that the cells are in a noisy state and maybe gradients are there to sort of superimpose the precision. Like we kind of have to kind of assume that are we assuming that most states are always, you're going to be jumping across various states and gradients are there to ensure that things are not going out of their way? I don't know. I guess I need your opinion, because it seems like fluctuations are necessary to make you jump from one state to another, or even to put you in one, kind of like the bet hedging effect in evolution. But I'm trying to see, how do we make it precise?
Maybe I'll try to say something to that. So there are various examples where cells move around on gradients, and there the fluctuation is a nuisance, right? Because you don't want, you want gradients. So for example, stiffness gradients are known to allow cells to migrate, right? And of course, chemotaxis is of course, dominant as well. So there, fluctuations are actually something you want to get over, and that the gradient allows you to do it, right? So that's sort of one way of doing cell migration. Here it looks like the opposite in some ways. That's why we found it surprising that you don't always need fluctuations, right? As I said, if you have a nice gradient, you just roll down and the fluctuation is just making things messy. Here it looks like an example. Again, it's, you know, it's going to be a bit controversial for sure. Here it looks like without fluctuations, you won't even have a migration because either the gradient is not strong enough in this case. It's not that the mouse embryo couldn't have evolved a gradient mechanism. That's not the argument. I think the argument is that some looks like this is how it works. So why, I don't know, right? That here it looks like so the later migration, as I said, once you form the stream, looks like the gradient is what allows the stream to move. But at this early delamination phase, it looks like if you don't have the fluctuations, you actually don't get anything, right? So they're not the something to get over, but they are the essential ingredient, looks like, at least in this very early part, right? And then you need some gradient, I guess, to trap things, if you will. And again, doesn't look like there's a nice energy type gradient. It looks like sort of an entropic type gradient where, you know, you move around and then you get trapped and you can't go back as efficiently. I'm sort of saying the same thing again, but I think just to distinguish that contrast, that here it's not that the gradient which is there and then the noise is kind of just a pain to get over, that it's sort of making things imprecise, but here it seems like the thing which is driving it, which again, maybe more needs to be done to set it. Okay, I'll stop.
Yeah, I wasn't entirely, I didn't totally see how you can be certain that it's the stochastics that is driving it. I mean, like how you can rule out that there is a deterministic effective pull via such a gradient. But I mean, generally speaking, I mean, certainly development wants to be precise, but at the same time, it's also always a question like how precise does it really have to be? So like I emphasize it can be very precise, for instance, in the neural tube, and everyone would say, yeah, you should be rather precise when making a neural tube, a central nervous system. But in reality, my colleagues tell me like, for instance, with motor neurons, we are making many more motor neurons than what we are using. So like once you're making, say, a limb, you know, all the motor neurons that are not connecting, they're just dying off. So there's anyway way more motor neurons than what you would actually need. And so, you know, you could really argue that you don't need that level of precision that we are now seeing, but we do see that precision. And so I think it goes both ways, really, that, you know, we at the moment focus on how you can get the highest precision possible, but we don't know whether you need it. And you, on the other hand, you know, you're focusing on the power of imprecision, of stochastic noise. And also there, we don't know whether there isn't a deterministic pull to really like somehow, you know, bring it back to order. And I think the answer is like, we need a lot more imaging and a lot more experiments to really be able to evaluate these 2 points. You know? But in both cases, the way I look at it, it's also very good to just make a catalog of what's possible under what condition, right? Because while we're busy getting data, we still need to know what mechanisms are there that could do one thing or the other, you know? And clearly you need some disorder in order to leave an epithelial layer. I mean, there's nothing more ordered than an epithelial layer. So like if you want to get out, you know, that's gonna create some disorder. But the question is how it's fueled, you know, what is driving that disorder? You know?
Right. No, absolutely.
Sid, do you know if there's a relation between the fluctuations that you see and changes in deadhesiveness? Like, for example, when you make the mutants that are more stable, quote unquote, is there like reduced cytoskeletal network changes? Is it just like more, because it's more stable, does that just mean that its adhesions are also more stable, basically? So
at least in this case, I should have shown some other picture as well. We didn't see any difference in adhesion proteins in counting. Again, that doesn't completely answer your question fully, but at least an attempt was made to do that. So here, I guess your idea is that maybe they're just stuck, right? So you increase addition rather than just decreasing fluctuation, and then you just make them too sticky. And I guess, I mean, the idea is, unless, knocking out myosin somehow upregulates addition, something like that could happen. But here it was a motor which was knocked out, right? And not all of them, right? Only one of them, for example. And from whatever measurements we could do on addition proteins, it didn't look like that their numbers were drastically changing in that sense. So yeah, maybe that's how I would answer it. But that doesn't completely rule it out, of course, like in these these contexts, right? But what we did measure is that, so the stiffness gradient, which has been seen in xenopus to drive this early part. So here we see that the stiffness, there's no stiffness gradient on the length scale of which the delamination is happening. Again, chemotaxis is still a relevant one, but at least in other contexts, people know stiffness gradient plays. That doesn't play a role here, we know. It plays a role later. You have a stiffness gradient, but much further away in the mesoderm. So once you have the streams, then they could sort of ride the gradient wave, if you will, right? There is a gradient waiting there. So once they reach there, a group of them, they can then fall, through that, use that gradient, but they have to sort of make it there. And there it looks like. So we don't know. Again, chemotactic gradients is another open one. We don't quite know. But the idea was that, usually, again, unless something like, as you said, adhesion is changing or something drastic is changing, reducing fluctuations would make things better if a gradient was driving things, and the fluctuations were just sort of, making things imprecise. So here it kind of looked like reducing fluctuations kind of enhanced stopped the migration, right? So you take out the fluctuation and nothing moves, basically, right? So the gradient, and again, as there's some interplay of these things. It can't be just one or the other, but you find examples where one kind of looks more dominant than the other.
And I guess on a higher, like related to your discussion with Dagmar on a higher level, I wonder if the fluctuations combined with like selection or something like that could help actually make development more robust, because then you can kind of take a broader set of cell states, I guess, and then if you, for example, make way more motor neurons, some of them might be kind of weird, but by producing more of them, you basically can pick out things that work, right? And then the embryo can be more robust to changes in it, whatever environment might be happening, or something to that effect. Like,
Dagmar, you have something to say on that? I don't think I have
I don't know. I mean, it's very difficult to comment. I mean, I don't know.
Yeah, it's kind of like what I said, kind of bet hedging effect. So in terms of evolution, the idea is you generate as much diversity as you can and eventually narrow down with some sort of selection process. I mean, I don't know development.
That's a better way to put it. My description is kind of mangled. But
well, the idea of bet hedging is you don't know what the environment or you're going to be in. I don't know if that's true for development, because you're
I don't think so, but it's
environment.
I would think it's very deterministic in development, but
looks very canalized.
On the other hand, it's also in development whenever something goes wrong, like for instance, if you're making a twin, right? I mean, so you suddenly in gastulation are splitting your embryo in two, and you're starting all over again, or like you're in the middle, but like you're only having half the cells, and just brings it back together because you have such a robustness in getting back to the right size before going through gastulation. And so, I really think that's why, when I started out as an assistant professor, one of the senior people didn't quite like the direction I was going. So like, go stochastic or spatial. And I was like, no, if I want to do development, I don't want to go to stochastic, because the thing is like, development is so robust and so deterministic that at the end of the day, stochastic effects, they will all be controlled somewhere, because otherwise you wouldn't end up with all, basically, if you have a different phenotype, it's because of a mutation. It's not because something randomly went wrong, and so that's basically why I really think that, you know, there's always like some robustness, some mechanism that actually controls for the noise. This doesn't mean that it wouldn't use fluctuations in certain places. So it's not going against it, right? But it's nonetheless, you know, I think that the phenotypes that we're observing are really are there because of deterministic processes. And that's the same, like when you look at patterning, I didn't talk about like, you know, these things like lung branching or whatever, where you have repetitive structures. It's very easy to get repetitive structures with stochastic mechanisms, but then you get repetitive structures that are different in every embryo. And so to have deterministic processes that give you reliable repetitive patterns, there's only very few mechanisms to do that, you know, and yeah, so I think that's, for me, development is so reliable, you know. So it's the opposite to bad hatching, I would argue.
Well, what if, this is just to instigate more discussion, but I mean, I sort of feel like, what if you have all the possibilities in development has evolved mechanism that controls? Like for me, the way I see it is anything is possible early on, because they talk about this chromatin loosening, all genes are available. And what if we have all the possibilities, because let's assume genes are all competing, but then different layers of mechanisms are, because evolution is different than development, right? Development progressed over an evolutionary process. So you got to sort of like separate these two. Let's say across an evolutionary developmental system, you generated these control mechanisms that over time, you know, overcompensated. Like one of them, the one I work a lot more in, is gene length and intron. The idea of introns being introduced into genes to delay. So let's assume all genes were competing all at the same time, and all of a sudden you need to introduce a gene later, but they all want to be introduced at the same time because it's available. And so one idea is to introduce introns into genes and delay when they show up. So, and I think, like, you know, we get these regulatory mechanisms and methylation to sort of put things in place. So it's not that noise is not there, it's just that development found a way to overcome noise. And this is why you get like these redundant processes. That make sense? So I could see why early on, maybe fluctuations are really important, you know, to shift you on from one state to the next. But then in order for you to maintain or to progress, you do need gradients or some other way to ensure that you are in the right state at that moment. That
yeah, or you could have you could have fluctuations that, for instance, drive you out of the epithelium, but then you need a mechanism that controls how many cells are coming out and, you know, shut it off, right? Something like that, because you need to control numbers or whatnot. Yeah.
But what about there are processes in development that are very stochastic, like, for example, neural development, cells finding their targets, where there's a whole selection that takes place, sort of analogous to natural selection, except that it's selecting targets and it's very, very stochastic. So how does that fit in?
Well, I think it does. It's the same thing. Like, you know, as I said, like no one knows why we have so precise, say, motor neuron numbers, when in the end you're only using like 70% or whatnot, right? This goes with what you're saying, because only those 70% randomly finds their target and the others are discarded. Then we don't know, is there something about the quality control? You know, is that basically that they just want to have the best neurons, or is it just whatever? But I think it's a very interesting and very, I mean, I've asked myself that, of course, right? And I think it's a very interesting question. I have no idea. I don't know, maybe.
If I understand you, you're pointing to the very predictable number of neurons you get, but then the exact branching structure and whatnot is unpredictable, right? So,
yeah, and also they're not all surviving, right? I mean, so you're generating those neurons, but they're only surviving if they find a target. And if they're not connecting, they just die off. So not all motor neurons that are born will survive. They will only survive if they're used, for instance, in the limb. And so you're making many more than what is used. And so, you know, and you're saying like, you know, it's a stochastic process, you know, the search process, and not all of them succeed. And so, yeah, but I have no idea why nature decided that this was the best way or its way of doing it.
I mean, immunology is another example where all the positive, negative selections define our immune system, right? And so clearly you generate a lot of diversity. There's the classic sort of, you know,
But that's hedge
difficult.
betting. I can see that one. But
Yeah, that is where I guess Maria's bet hedging example goes. And I guess in embryos, not embryos, sorry, in reproduction sort of things happen, right? Like in C. elegans, egg formation, you can see lots of eggs dying and things like that.
same in humans. Same in humans.
Same in humans, probably, yeah, which I don't know. So there, I can imagine sort of the strategies of, you know, weeding out things which are not going to work out. But why do you generate them? Why don't you control generation of them? Maybe relates to things like you want to generate some diversity. So there could be examples where it kind of comes in, I guess. Yeah.
But it's maybe, yeah, indeed, it might be a quality control that neurons are so important. You need that level of quality control.
You just, yeah, you want to throw as much resources as you don't care about the resource, and you just, yeah exactly, want to have a
very good point. Yeah.
My lab is really interested in these questions of kind of both stochastic but more deterministic competition in early development, both in mouse and human. And I always found it so remarkable how much selection there is, at least for human embryos, like even just that implantation stage, such a high degree of filtering. And I'm not sure how high that is for other organisms and why a kind of evolution has played out that way for humans, but I think it's really interesting.
Yeah, I mean, human implantation is like a massively invasive process where everything is, you know, stacked against it. I think like it should an implant, but then it now, you know, yeah, exactly.
And also the high degree of like mosaicism that occurs in human embryos and how that corrects itself, and that robustness is kind of there to perturbations. It's really interesting. And I don't think we understand those mechanisms completely, but we're interested in the sort of competitive interactions between cells that regulate it.
But do you think it's mechanisms that control it? It's really just competition, survival, and that dominates there. Whoever survives is kind of survives, or is it? It's sort of.
Well, I think it's a combination of both, right? There seems to be some emerging evidence that cells are kind of regulating each other and kind of keeping an eye on their neighboring cells and kind of triggering elimination in a way, although the exact molecular mechanisms of how that works is not clear. But there seems to be some sort of sensing and responding to your environment at that early stage. And that's one mechanism by which these kind of aneuploidic cells could be purged.
Cool.
I guess it makes like evolutionary sense for humans, at least, that there's a high filter for that implantation step because it requires so many resources to actually develop the embryos. If you pick something that's not like suboptimal quality, then it's a lot of wasted energy, you know, so it's not evolutionarily advantageous.
Yeah.
I actually had sorry, go ahead.
No, but it's also a crazy process, right? I mean, hatching, I mean, like you wouldn't design embryogenesis this way if you were to engineer it, right?
I think that's probably true of a lot of biology. It's like, why is it this way?
But I agree. I agree. It's a good point to select. Yeah.
I actually had a question for you, Dagmar. Is there like a minimum size for the slimness of the cells? Because I was imagining like it's advantageous for it to get smaller to improve precision, right? But then why don't they just become like super tiny? And
yes, I think the limit is the nucleus, because I mean, you could, of course, given that you're pseudostratified, you could make it smaller and smaller. I'm sorry about the telephone. It's my parents. I'm in my parents' place. But so it's the apical surface could get smaller and smaller, but at some point, like, you know, if you're making it too small, you don't have the mechanical stability of an epithelial layer because you still need to have the adhesion belt and everything. And effectively what Sid was describing of like the cells, you know, going out of the epithelial layer, like if you're making that adhesion belt too tiny, the cells would just basically eliminate from the apical surface, right? They would just basically not be able to keep their contact on the apical surface. And so I think that's the limit. We see this in simulations, and everyone else in the field, even if they're not spelling it out, if you have any kind of vertex model or any other, you know, cell-based model, once your cell surface becomes too small, everyone has some rule like that. You have to eliminate it from the layer because numerically it would cause problems, but you can be certain it would also cause the same problems in biology. So I think there is just a limit as to how small you can make it, given, you know, effectively your bending stiffness of the adhesion belt and everything. You know, at some point you can't support it anymore. But we have wondered what this size might be, but it's effectively you never see cells that are going below, like say 1 or 2 square micrometers in surface area. And so that seems to be the limit. And that's what you're seeing like in the eye disc. If you're going close to the morphogenetic furrow, where you have the decapentaplegic source, the DPP source, you know, you're getting really tiny surfaces, but I think they're about 1 square micrometer, and that's it. And these cells are really weird if you're looking at an eye disc. I think that's the limit.
Actually, it made me wonder, what if like the early fly embryo, you know how they actually are almost like one cell with many nucleus? Maybe the reason it's like that because they couldn't get any smaller.
That's a good point. We didn't argue from this direction yet that the syncytium is really like the ultimate case, right? I mean, where you just have the nuclei and yeah, maybe.
But zebrafish doesn't do that, right? Zebrafish keeps making very, very tiny cells, right? I don't know what size is there actually. At the early part of zebrafish development is you have this big egg, one cell, and just make tiny, tiny, tiny. It's like really gets like, I don't know, several thousand cells and very quickly.
Yeah.
Because they're not making new stuff, they're just making membranes, which has always seemed odd at some level. But
If
And
you're,
right? Like,
Sorry?
like most embryos early on, they divide more and as opposed to grow, like even the mammalian.
yeah. But they make membranes unlike Drosophila, right? They actually make
Right.
cells, tinier and tinier cells rather than syncytium, right?
and if you're referring to the epithelium on the zebrafish, that's actually a squamous epithelium. So it's kind of the opposite architecture.
I see. Okay, I don't know enough about.
Long and flat.
Yes.
I see. I
Yes.
see. Okay. Cool.
I know we're quite over time, but this is great. Like, I don't want to stop this, but I want to make sure that anybody who needs to go knows. I think this is the most enjoyable part of the discussion, is be able to join both perspectives. I hope everybody has enjoyed it.
Both very interesting talks. Thank you.
Yeah, thank you.
I just want to say I put a lot of the papers that I followed that you showed, Dagmar, into the spreadsheet, and I also tried to put them in the talk, but I couldn't find the ones that you showed, Sid. So if you don't mind,
Oh, that's bad. Okay, I'll send it to you.
especially the ones like from Savan, I just couldn't find it. I don't know what's going on, but here I put them here in this Google
Pasted them this
spreadsheet.
morning.
Oh, okay. So maybe I just Googled, I did something wrong. I just
That's
meant
fine.
like before you go, I just think everybody can access these papers if you don't mind. So there's a papers spreadsheet in this Google shared sheet that I try to maintain. And so on the bottom, I tried to put as many of the papers as I could find. If you don't mind adding
Yeah, do
that I missed.
So what was this? Is this all the papers that Dagmar mentioned, like all the
So-
other work? Or?
Yes, so I don't know if I got all of them. I have to actually go through the paper again, but I thought maybe if Dagmar sees the ones I missed, you can add, or if you see anything else
I
retaining.
noticed
'Cause I
that-
just feel like if somebody's gonna watch the video, then you can also check
Mhm.
them out, and I tried to find them
I
along the way.
noticed the
So
cumulative-
they are in the videos then, I see, okay.
Great.
So I am recording it, and that's the other, actually we can stop now, so just to